and welcome back to your YouTube channel, AgriAddict. I'm your mentor for this crash course on horticulture. My name is Dr. Deepthi Velaturi. I've been graduated in horticulture from Acharya Engineering Agriculture University, Hyderabad. I've done my master's from GKVK, University of Agricultural Sciences, Bangalore, in the Department of Fruit Science. I have also completed my doctorate from Dr. YSR Horticulture University, Tadepalligudem, Andhra Pradesh. During my academic journey, I have achieved All India rank 21 in ICR AIEEE PG entrance exam during the year 2005. I have also cleared my ASRB net for the years 2008 and 9. Let us start our discussion on the second lecture, that is methods of propagation of horticulture plants. So let us understand the term propagation. Propagation, plant propagation refers to the multiplication of an individual plant or group of plants which have specific value to mankind. That is either for agriculture or horticulture, that is raising of plants which are of useful for mankind. So the plant propagation is multiplication of plants which are useful for mankind. And simply we can say that perpetuation of plants is called propagation. The term perpetuation means existing. Existing on earth. For the existence of earth, plants need to multiply on a large scale. But if you see in agriculture, sexual reproduction or by seed is the dominant method of practicing new plants. Whereas in horticulture, the case is different. Both sexual as well as asexual reproduction are common methods of producing new plants. Thereby, these new plants or individuals are required for establishing new plantings, new gardens, and new orchards. But if you look into the exact definition of propagation, it is the art and science of multiplication of plants. So it required skill. The propagation methods can be broadly classified into sexual, asexual, and micropropagation. Let us go one by one into the details. The first one is sexual method of propagation. What is this sexual method of propagation? This is raising of the plants or the multiplication of plants by means of seed. How is this seed formed? Due to the fusion of male and female gametes within the ovule of the flower. So here you can see a normal plant which produces flower which contains both male as well as female parts. These in turn produces male and female gametes, and the fusion of these gametes which occurs in the ovule of the female part. So finally, the fusion will leads to zygote, and the zygote will transforms into embryo that is present in the seed. Plants that are produced from the seed are called seedling. The seed is made up of three parts. The outer coat, here you can look the transverse section of the seed. The outer part is called as outer covering, we call it as seed coat, which protects the seed. Then the endosperm. So this is the food storage. It is the food reserve for the embryo. So embryo, and the third part is the embryo itself, which is the egg plant itself. So upon germination under favorable conditions, this is going to germinate, and then again the plant is formed. This is the general life cycle of a plant which produces seed, and the multiplication or the propagation for commercial cultivation is through seed in most of the agriculture as well as horticulture plants. Horticulture plants, especially vegetables and flower crops, are the main propagating material is seed only. Let us look into the advantages as well as the disadvantages by seed propagation. The first one is evaluation of new varieties, or by breeding works, the hybrids are produced from seed only. This is the major important point. Then in terms of horticulture, if you see, the propagation of root stocks is by seed propagation only in order to have better root system. Then viruses don't transmit through seeds. Thus, mostly seedlings are free from viral diseases. Then the fourth one is seeds also offer convenient method for storing of plants for a long time. So we can germinate them as and when required. So we can save them for the next crop until the next crop is being followed. Then the disadvantages are also there. If you see, seedlings have a long germinal period, especially in case of perennial plants like fruit crops. The bearing or the fruit bearing comes later when compared to sexual seedlings. Due to segregation of characters, the progeny is not true to type. So this true to type is very much important in case of horticulture plants. So in order to get uniformity in growth and production of produce. Then the third one is it is not economical. So to handle large trees, as large number of trees can be accommodated per unit area, and the cultural operations become difficult if you are going for raising of nurseries with the help of seed propagation. The seedlings usually show vigorous nature in terms of perennials. So it is very difficult. The cultural operations like weeding, other pruning, training and pruning techniques will become difficult in case of perennial plants like fruit crops. So by looking upon these disadvantages, it is always better to go for the second method, that is asexual method of propagation. Let us know what is meant by asexual method of propagation. This is the propagation of plants through any vegetative part. It can either be called as vegetative or asexual method of propagation. So any vegetative part of the plant, that is like leaf, stem, root, and so on. So it need not be necessarily through seed only. So the plant can be multiplied into a number of plants, and the progeny by this method of propagation are called clones. Just remember this word, which these clones are identical in genotype with the single plant, source plant, from where the plants are being multiplied. That is, we cannot call it as mother plant also. Then there are certain set of advantages as well as disadvantages for this asexual method of propagation also. Let us look into this. So some fruit plants like banana, and pineapple, which do not bear seeds naturally. So the term we call it as parthenogenesis. So they are, parthenocarpic. These fruits are parthenocarpic in nature. So we'll deal this in the coming lecture. These plants, banana, pineapple, and fig, and some other plants, fruit plants, they doesn't bear seeds. So for this commercial method of propagation is only through vegetative parts. Then the plants, there's a second advantage is plants are generally true to type. So there is no segregation of characters because we are not following the seed propagation. Here any vegetative part is used for multiplication of plants because these clones are uniform in growth, yielding capacity, as well as the fruit quality. This is one of the major advantage. Then plants have restricted growth. They are not so vigorous when compared to seedlings. And cultural practices are obviously easy. Then the fourth advantage is advantages of rootstocks can be obtained. So in the coming lecture, you can go with the details of budding and grafting. For susceptible varieties, on resistant tolerant rootstocks. So this also holds good. Then they have short juvenile phase and they have long bearing phase, early precocity. All this can be taken care of. Then the disadvantages are also there. The new variety cannot be evolved by this method. So because all the plants or the plants which are obtained from the mother plant are true to type, so we cannot obtain the hybrids or varieties by this method. It is only possible for commercial cultivation. Then the plants are not so vigorous. They are not long-lived. If they are not so vigorous, they cannot be long-lived. Then they can transmit viral diseases from plant to plants, especially in case of micropropagation. We'll go into details of this in the coming slides. Then the fourth one, that is also major disadvantage. Then the fourth one is vegetative propagation. In many cases is more expensive when compared to seed propagation. Seed propagation is a natural way of multiplication of plants. So the plants generally produce seeds. But if you are going for vegetative propagation, it is with the help of labor, that too skilled labor is required for some methods of vegetative propagation. So obviously it requires much inputs. See, here is the flow chart which shows the vegetative propagation in plants, either vegetative or artificial. So this is man-made. So vegetative or naturally the plants also reproduce, also multiply by means of parts of the plant. For example, if you see, the vegetative part can be broadly classified into three different divisions. The first one is propagation by adventitious bud. The next, the propagation by terminal bud. Then the propagation by stem. So these two are of not commercial horticulture importance. So they might be present in other groups of plants. So the propagation by adventitious may be of root bud, stem bud, or leaf bud, but we are not concerned here. The only concern of natural propagation is by stem. That too, the stem can be of three different categories, like the underground modified stem, then the subaerial modified stem, then the modified aerial stem. So under this underground aerial stem, we have four different variations. The first one is tuber, rhizome, bulb, corm, and subaerial modified stem. So underground modified stem is the stem is modified below the soil. Subaerial, it is somewhat underground and somewhat on the top of the soil. Under this, there are some more deviations like runners, suckers, stolons, offsets. Then modified aerial stem. The stem itself, the stem of the plants of different species itself is modified for multiplication of the plants. So either I'll tell you in details. This may be of either stem tendrils or thorns or phylloclades or cladodes, bulbils, pseudobulbs, and thalamus. So all this comes in one category. But if you look into this comes naturally. If you are going for commercial propagation, as I told you, propagation is the art and science of multiplication of plants. So we are multiplying artificially. We are doing it manually. So if you look into the different divisions of this artificial vegetative propagation, the first one comes is cutting. Second one is layering. The third one is grafting. Further, it can be scion grafting or bud grafting. So in general, the scion grafting is called grafting itself, and bud grafting we call it as budding. So under this cuttings also we have root cuttings, stem cuttings, and leaf cuttings. Layering can be of different types, whereas budding and grafting can be of different types. Now we'll go in detail. So as I've told you, these are the specialized structures. So natural vegetation propagation by vegetative structures are present in the plants itself. So these are the certain plant modification which are used for vegetative propagation of plants. The modified plants of parts may be stem, root, or leaves, which are usually specialized for food storage. But they are able to develop into individual plants. So that is the reason why this is a typical mechanism for the plants to multiply as well as store food. So basic two principal methods are used for propagation of the plants by using these modifications. The first one is separation and the second one is division. So the naturally detachable structures. So plants have a mechanism to store food and to multiply the stored food, and they can further be developed into individual plants. So they can be easily detachable. So the human intervention is not required. The examples here are bulbs and corms, which can be separated and then planted individually. Then the other one is division. The plant modifications such as rhizome, tubers, and so on, etc., can be cut into sections. So this has to be done manually to obtain new plants from each section. So let us understand the examples. Here, the variations of the plants. The first one is runner, that is strawberry. Then bulb, we know it is onion. Then corm, gladiolus. Rhizome, iris, ginger. Then tuber, potatoes. Tuberous root is sweet potato. Offshoots, banana, pineapple, and so on, date palm. So let us go into one by one. The first one is bulb. Bulb is a specialized structure. Here you can notice the picture of bulb containing a short vertical stem axis or the growing point enclosed by thick fleshy leaves. So here is a bulb. The growing point is here, and these are the scales. Thick. These are thick. You can imagine the onion, so which is of layers, different layers, out of which the apical point or the tip, growing tip, is being closed. So it being protected with the help of these scales. So these scales are called bulb scales. Morphologically, the sheathing leaf bases. So upon which this is underground, upon which the plant grows. So the leaves are visible, but however, the leaf bases are swollen, and these are in the form of a bulb. So the name bulb is formed. Then the growing point develop in the axils of these scales and produce miniature bulbs. Here you can see upon this leaves bases also, you can see some more growing tips are being produced, and after a due course of time they will be separated, and again leaf bases will be loosened. So you can simply detach this bulbil, and then this can be used as a propagating material for the second crop. So these points develop in the axils of the scales and produce miniature bulbs, and these are also known as bulbs or bulblets or daughter bulbs. These can be separated and the mother plant growing season, and then use can propagating material. Examples here are flowers like tulip, daffodils, tuberose, and vegetables like onions and garlics. In garlic, each bulblet is called as clove. Then the other variation is comb. Here you can see the comb. A comb is a solid underground. This is also underground base of a stem which has nodes and internodes. There only scales. We can found scales or the two leaf bases. But here the stem which have nodes and internodes are condensed to form a dry scale like leaves. So this is also somewhat similar to bulb also, but it contains of dry scales like leaves. After flowering, one or more combs may develop. So after flowering of the, if for example, if you see gladiolus, after the finish of the flowering phase, then more combs may develop just above the old one. So here you can notice small bulblet, which disintegrates. In several new combs and cormels, these are called cormels, develop below each comb. This may be separated and grown for 1 to 2 years to reach the flowering stage. Example are gladiolus and am. So upon separation of these cormels, they can be used for propagating material for the next crop. Then the other deviation is tubers. So we all know the example is potato. Here you can notice the potato tuber, which contains the eyes or the swollen or the sunken spots, where a bud is present. So the tuber is also an underground stem which has become thickened due to accumulation of preserved food material. Propagation by tuber can be either carried out by planting the whole tuber or by cutting into section, each containing bud. So these sunken areas will contain bud, and then upon favorable condition, they started sprouting. We can cut them and then use further as propagating material for the next crop. Then the next deviation is tuberous crop. The examples are sweet potato, dahlia, tapioca, or cassava. Then these plants produce thickened roots which contain large amounts of stored food. The tuberous roots differ from tuber. Why? In case they lack nodes and internodes. So the nodes and internodes are present in tubers, but they are lacking in tuberous roots. The adventitious buds are present only at the stem end or the proximal end. Here you can see these buds are only concentrated at the distal end or at the proximal end. Fibrous roots are produced towards the distal end. So from the base, fibrous roots are produced. These fleshy roots are separated and used for propagation. So you can cut up to here and then you can use it for the next generation, next crop. Then the other deviation is rhizome. The horizontal, thick and fleshy or slender and elongated stem. So they either be horizontal, thick and fleshy, or slender and elongated stems. Growing, here you can see, they can be thick or slender. Growing underground, these are also underground storage structures known as rhizomes, especially in case of banana, ginger, ferns, turmeric, cardamom. All these examples are rhizomes. These rhizomes also have nodes and internodes like tubers and readily produce adventitious roots. So adventitious roots are present like tuberous roots, where the nodes and internodes are concentrated. The rhizomes are then cut into pieces containing each vegetative bud and then transplanted. Here, the buds are also present, the nodes and internodes, and the fibrous roots are also present. So by cutting them into pieces and utilizing as crop for the next season. Then runners. Runners example is strawberry is the best example. These are the specialized aerial stems. Up to now we have seen underground stem. This is the aerial stem which is called as stolon. Here you can see this is a connection between one crop to the other, one plant to the other plant. So these are the aerial stems, specialized aerial stems called stolons, arising from the leaf axils of a plant which has rosette crowns. So these are the rosette plants. So nearby on the whorl, these leaves are present. So from there, the fibrous root system is allowed, and then the stolons, the aerial stems, the specialized aerial stems will grow, and then upon touching the ground, they again start producing fibrous roots. So upon division, we can divide and use this as a new crop. From these runners, more new runners may arise by developing a natural clonal propagation method. Then the other deviation of horticulture importance is suckers. Adventitious, the main important example is banana, chrysanthemum and curry leaf. The adventitious shoot from the underground portion of the stem or from their horizontal root systems are known as suckers. Here banana has the rhizome. You can see this picture, the rhizome, upon which the suckers are being produced. So more number of plants will arise, but being the main single plant for the growing season, which produce the fruit. Apart from this, we have sword suckers, water suckers in case of banana. And if we cut the rhizome part, the underground part, and this can be used for further propagation. So the suckers, they strike roots and they may be utilized as propagation materials. Well-developed suckers are dug out and separated from the mother plant and planted in nursery for further growth. Because the underground modified stem is rhizome, so it develops a fibrous root system where the nodes and internodes are present, and each bud on the rhizome will develop into a new sucker. So we call it as sucker. From the mother plant side by many suckers are developed. So this can also be utilized for propagation, for commercial propagation also. Then the other important variation is offsets or offshoots. The best examples here are date plum and pineapple. Date palm and pineapple. An offset is a shoot or thick stem of rosette-like appearance. So nearby, so many branches arise at a one single point. So that is called rosette. Here also you can see one single stem, a condensed stem, where the leaves and the leaf bases are being grown at the base of the main stem in certain plants. The commercial propagation is by separating the offshoots and replanting them again. So here you can see the picture of date plum, which produces offshoots, and these can be separated and then planted again. Here we have a separate term called slips. So this is also a rosette like a sucker or offshoot, which is called slip in case of pineapple. And here the offshoots can also be called as suckers, aerial suckers and ground suckers. Some of them can arise below the ground, so they call ground structures, and above the ground we call it them as aerial suckers or offshoots. Until now, we have seen the specialized structures. Now we'll go in depth with the different vegetative propagation methods of horticulture importance one by one. The first one is cutting. What is this cutting? Cutting is a method of asexual propagation in which plant part such as stem, root or leaf is cut from the parent plant and placed under favorable conditions to form roots and shoots, thus producing the plant into an independent plant, new plant. So just simply cutting the stem or root or leaf which are placed under favorable conditions. So they may give rise to a new plant. This is as simple. Then the advantages of horticulture importance are, it is the cheapest method of asexual method of propagation. So out of all the different methods of vegetative propagation like cuttings, layering, grafting and budding, before we have seen in the flow chart. So among all this, cutting is the cheapest method of propagation. Then it is used for clonal multiplication of rootstocks. So one more advantage is for the rootstocks in case of budding and grafting, cutting is the best method for propagation. Then the types of cuttings can be broadly classified into stem cuttings, root cuttings and leaf cuttings. Here I can see the stem cuttings can be classified into hardwood cuttings, semi-hardwood cuttings and softwood cuttings. So the hardwood cuttings, let's go into the details and examples. Here is a table which shows the different types of stem cutting, age of the cutting, size, and then examples. So in case of grape, pomegranate, figs, pear, plum, all the fruit crops of temperate and tropical, some of the some of the flower crops like bougainvillea and poinsettia, in those cases, the hardwood cutting of 1 year old and the size of 10 to 30 centimeters long and 1 to 2.5 centimeter dia is induced to form root and shoot. So the cuttings of 1 year old, here you can see the hardwood cutting would be like this. It has a thick dia stem with so many buds. So this is the best commercial method of propagation for these crops. Then say the second type of cutting is stem cutting is semi-hardwood cutting. So which is around 5 to 9 months old and we at least 10 to 15 centimeters long or 1 to 2 centimeter dia. It is thinner when compared to hardwood cutting, and this is practiced in case of citrus, olive, camellia, some of the croton crops, herbaceous foliage plants, then roses, poinsettia, bougainvillea and all this. The commercial cultivation is semi-hardwood cutting. Then we can go for softwood cuttings, especially these are 4 to 6 months old. They are still small. We have to take at least 7 to 15 centimeters in length and 1 to 1.5 centimeters in dia. So this is most commercial in case of foliage crops as well as flower crops. Horticulture, magnolia, oleander, justicia, maple and juniper. Then the last type of stem cutting is herbaceous cutting, just 2 to 3 months old, and there is no specification for the size. This is very good for propagation of coleus, carnation, geranium, flower plants, chrysanthemum. In case of tomato also. Then among the fruit crops, pineapple and vegetable, sweet potato, herbaceous cutting also holds good for commercial propagation. Now we have seen the stem cuttings. Then other important vegetative propagation is by leaf cuttings. So this depends on the species, on the whether it is a whole leaf blade or leaf blade sections or the leaf with the petiole is used. So leaf cuttings can be classified into blade cuttings. Example, sansevieria. Here you can see the leaf blade can be cut and then planted in the soil to form a new plant. Then vein cuttings, example is begonia. So here you can see the veins if they are cut, then the new leaf or the new plant arises, small plant arises, which can be detached and then planted in the soil to form new plant. Then leaf bud cutting in case of blackberry, camellia, lemon, rhododendron. This is also of horticulture importance, commercial horticulture importance, raspberry and all. So with the petiole, if it is cut and then planted in the soil, so upon favorable conditions, they start producing roots, and then it transforms within a course of time. It transfers into a new plant. Then the other deviation is leaf margin filler. So leaf margins, when they are cut, they from there only new plant arises, which can be taken and planted in the soil. Then the other vegetative propagation methods, apart from cuttings, it is layering. So what is exactly layering? Layering is the development of roots on the stem while it is still attached to the parent plant. The root system is then reached to become a new plant growing on its own roots. Such root system is called layer. We have different deviations under this layering. The first one is simple and tip layering. So the tips of the shoots are buried 5 to 10 centimeters deep into the soil. In case of tips, whereas in case of simple layering, just the shoots are bent and then the tip is exposed above the ground. But in case of tip layering, the shoot is immersed into the soil. So that is the only difference. Then rooting can take place in the current season shoot, which is bent to the ground. The examples are given in the next slide. So let us go into the details of the next method of layering, that is trench layering. The growing plant or the branch of the plant is kept in a horizontal position from the base of a trench and filling it with soil where the buds are present actively and new shoots may develop and the shoot bases are mutilated. So they are not given sufficient light. So thereby the roots also develop from those places. Hence we can cut them and use into new plants. Used for commercial cultivation of new plants. The third one is mound layering also. Stooling. This is the simplest method for Sion varieties. Mound layering is mostly for most of the fruit plants. So the plant is headed back and then above 15 centimeters the ground level during the dormant season, especially in case of temperate fruit plants. Then it is allowed to put forth into new branches where the branches also produce the roots, and then upon removing them they can be made into a new plant. Then the other deviation is serpentine or compound layering, modification of simple layer in which 1-year-old branch is alternatively covered and exposed. The exposed part of the stem should have at least one bud to develop new shoot. So the serpentine layering is alternatively. So the shoot of the plant is being put into the soil at some places and above the soil aerial, and again put into the soil and again aerial. So the plant part, the shoot part which is produced, which is underground, started producing roots first and then shoots. So upon cutting this connection, we can directly make the plant into make it into an individual plant. Then the last method of layering is of commercial importance, horticulture importance, is air layering, is also called goutte or marketage or pot layering or circumportion. So this is first introduced from China. So you have to remember for this exam point of view. Then the moist rooting system, rooting media, that is the sphagnum moss is being used here, is wrapped with the help of small polythene strip of 200 to 300 gauge. So the main process is called etiolation. Etiolation means shutting down of light, that is showing darkness for the shoot, and upon moist rooting medium, the shoot of the plant start developing roots. Thereby, when it is detached from the mother plant and planted in the soil, it can develop into a new plant. Rooting generally commences from 25 to 30 days, and the layers are ready to transplant, and the plants produced from this air layering are called layers, transplanted within 3 months. So after 1 month, we can achieve rooting. Upon removing and planting them, the plant will be ready for about 3 months for commercial cultivation. The air layering can be practiced in the month of February to March or July to August. Here you can see the picture which is showing different types of layering. As I told you, the tip layering, the tip of the shoot is put underground, and thereby the rooting can be observed. After rooting can be after formation of new shoots, then it is detached from the mother plant and can be used as a new plant. Then in case of simple layering, so the tip of the shoot is exposed, only the part of the shoot is underground. Then in case of trench layering, a small trench is dug out between the sides of the mother plant, on either sides of the mother plant. So the branches or the shoots can be etiolated, fully closed, and the tip is exposed, fully closed under different areas where the bud is present, shooting as well as rooting can be observed. Then serpentine layering is similar to trench layering only, but some of the part of the shoot is underground and part is aerial, and again some of the part of the shoot is kept underground, and then it is left alone aerial. The place where it is left, it is kept under the soil, it starts showing rooting. So thereby an individual plant can be obtained. Then the mound layering, here the plant is, as I told you, it is cut back to about 15 centimeters from the ground, and then it is covered with soil. So after a few days, we can notice different branches or shoots putting forth from the plant, and individual shoot will have produced roots also. So thereby detaching individual plant can be used as a single plant. Then aerial layer. So this is directly done on the fruit plants. So part of the shoot is being etiolated. So it is exposed to darkness. Then thereby we can achieve rooting. Here you can see the rooting, and upon cutting here, removing from the mother plant, directly it can put in into the soil for the next crop. The examples of tip layering are raspberry and blueberry. Then simple layering is followed in case of flower crops like bougainvillea and jasmine. Then trench layering is mostly followed in case of temperate fruit crops like apple, pear, and walnut. Serpentine layering is followed in grapes. This type of layering is followed in grapes for commercial production. Then mound layering is seen in Goa. The best example is Goa. Then to some extent for obtaining rootstocks of apple and pear. Then air layering. Air layering is followed in tropical fruit crops to maximum extent, that is lychee, jackfruit, Goa, and cashew nut, acid lime, and some of the foliage plants like crotons as well as ficus species. Before going into the grafting, I would like to announce you that we offer 2 sets of pre-mock tests, which is accessible in our app or website. You can make utilize of these tests to enhance your JRF preparation. But if you are seeking for a comprehensive mock test on a syllabus for the entire syllabus, you can go to our mock test. You can enroll for our mock test bundle, which is available for $299 only. Then we also offer online master course which includes video lectures, study materials, mock tests, previous year question paper discussions, and also doubt clearing sessions. The links of all the courses are given below in the description box. Click the link and register today. Then comes the method of grafting, which involves both the scion grafting and bud grafting, and which can be called simple grafting or the simple budding. So grafting is nothing but the art of uniting or joining two parts. So it is a art of joy. This is a skilled technique which involves human intervention. So it is an art and science of joining and uniting the parts of two independent plants in such a manner that they unite together and develop into a single independent plant. So on a whole, so we are uniting two different plant parts to form into an individual plant. So the part of the plant which is used as graft to become the shoot system is called scion. The part of the graft or the part of the plant which is used for the root system, we call it as rootstock. So these are the terms which of commercial importance. Then the interstock, then the other term is also there, interstock or intermediate stock, which is used for some species to overcome incompatibility. So the unity between the scion and the rootstock, if it is good, then we can form into an individual plant. If the rootstock and scion are not compatible to each other, then the unition it will not form. Then the joining will not form. So in that case, interstock plays an important role. Let us look into the factors which influence the success of grafting of scion and rootstock. These species, the scion and rootstock species, should be closely related. Then only the union will occur. Then they should have equal thickness. Then the scion and rootstock are of 1 or 2 years of age. They should not be more than that or less than that. There is close contact with the cambial regions of the rootstock and scion. So the xylem and phloem, if we cut open the stem of the scion or the rootstock, they should exactly follow the exactly put at the place where the cambial regions coincide, whether it is xylem or phloem. So the exact place it has to be united. Then the cells at the cut surface of the stalk and scion should not be desiccated, though they should not be dried. They should be immediately after cutting of the rootstock or the scion from the mother plant. It is required immediately to attach them and then cover them, so that the success of grafting is more. Then if we look into the details of characteristics of ideal scion, we have for ideal scion as well as good rootstock, one should keep into mind when we are going for either budding or grafting. Then the mother plants of the variety should be of genetically true to type. The plants should be healthy, free from any diseases or pest infestations or even physiological disorders. So abiotic, and it should be free from abiotic and biotic stresses. Then the plant should have known pedigree records regarding the potential bearing, fruit quality, and problems, if any. So for the past record should be there for the past 5 to 10 years or so on. So the mother plant should holds good the bearing potential, fruit quality, and other. Because this is a scion which bears the fruit. The plants should be of precocious and prolific bearer. Prolific bearer. They should be precocious, like early bearing. Variety should be there. So we need to be careful while selecting the scion. So the scion should contain the mother plant should contain all these characteristics, where the scion stalk, the scion bud or the stick is selected. So the scion stick is selected for grafting and the scion bud is selected for grafting. So the mother plant should contain the necessary qualities in order to select as a scion. Then the characteristics of the rootstocks. If you look, it should exhibit, the first one being, it should exhibit high degree of compatibility with the Sion cultivars. So we should know whether this will fuse with the Sion cultivars or not to give maximum yield or productivity of the trees. Then it should be well adapted to agroclimatic conditions like drought, frost, cold and heat, because this is the this holds the root system. The rootstock should be is directly in touch with the soil, so it should be at least tolerant to these climatic conditions, adverse climatic conditions. Then it should be resistant to diseases and pests for that concerned area. Then it should be tolerant to soil adverse conditions like salt, salinity and alkalinity. So we have to look into these characteristics in case of Sion as well as rootstock before going into the procedure. Here is a table which shows the different rootstocks for different fruit crops which are under commercial cultivation. So the apple dwarfing rootstocks, especially the apple tree, when they are grown by seed or any other type of propagation, it may show vigorous growth which is not required on a commercial scale. So for that dwarfing rootstocks, they put down the growth of the Sion, and they bring the plant into early bearing, so precocity, so that the cultural operations, if the plant is dwarf, the cultural operations is also easy to do. And here you can see the term M. This is of Malling research station. They have released the dwarfing rootstocks in apple, and these rootstock like M9, M27, M111, M106 are commercially used rootstocks for grafting in apple. Then comes Ber. The species can also be used for as rootstocks for Ber. So Josephus jujuba can be grafted. The Sion can be grafted on Nummularia, Josephus rotundifolia. In citrus, other species can be grafted, like Rangapur lime can be grafted for sweet orange varieties, Karnakata, rough lemon, trifoliate orange, flying dragon. So these rootstocks are very much important. At least you can expect 1 or 2 questions from the examination. Then grape, Salt Creek, Dogridge, Rapario Temple, these are tolerant to salinity. Please prepare a chart so that which rootstock is susceptible or tolerant, resistant or tolerant to various abiotic or biotic stresses. Please know, please underline, and please remember the crop and also the rootstock. Likewise, we have guava, the other species like Friedrich Thalianum, Pumilum, can be used as rootstock. Like in case of mango, the different cultivars like Korukan, Olar, like Columban, then they are resistant to salinity to some extent. Then Sapota, Kirani, the other species is used as rootstock. Then in case of walnut, Juglans, Paradox is a hybrid between Hindsey and Reacher. This is largely used as a rootstock for walnut cultivation. Then in fig, Ficus glomerata is used as a rootstock. Then if you look into the different types of grafting, the grafting can majorly be divided into attached Sion method and detached Sion method. So the Sion is attached to the mother plant. So the different methods under this are approach grafting, so which is the commercial method of propagation for mango. Then detached Sion methods are the different types of methods. Here we have mentioned. These are the main 2 methods of grafting. Well, if you look in attached methods of Sion grafting, the Sion is still attached to the mother plant, as I told you, till the graft union takes place. Whereas in detached, the graft of the Sion is separated. The Sion is separated and from the mother plant just before grafting. Whereas the roots are still intact with the mother plant, the Sion is separated. So, but in case of approach grafting, here you can see 2 different plants. This is example of splice approach grafting. They are cut as a they are sliced. The roots is the stems of each the plant is cut down to a slice, and then which the cambial tissue is exposed, and then upon bringing them together, we can tie them with a rope, then we can achieve the unity. So this is one type of grafting. And approach grafting, the root system is taken to the mother plant, that is to the scion. After unition, then it is cut back, and then this is commercially propagated as a individual plant. So I can give the examples also in the later on. Here you can see approach grafting and the different types of grafting: approach, veneer, whip, cleft, wedge grafting, inarching, bridge grafting, epicotyle grafting. So the grafting on the cotyledons of the some of the fruit crops. So epicotyle grafting is also important in case of mango. Then softwood grafting and top working. Here if you see cleft grafting, bridge grafting and top working can be used for old orchards to rejuvenate them with new varieties. So the root system, the trees will have the old trees will be headed back, and then the root system is still intact, and the headed back trees can be rejuvenated with the help of this top working with new scion cultivars. Here is an example of whip or tongue grafting. So it is cut into W shape so that both rootstock as well as scion are cut into the same shape, and then inserted, then tied, and the union takes place. Then the different pictures we show the different varieties of grafting. The other one is veneer grafting, which is also popular in mango. Here you can see it is cut in the shape of L, and then on the rootstock, and then the scion is also cut on the sideways and then inserted. So this is side grafting. This is a variation of the side grafting in case of mango. Then you can see the epicotyle grafting. So as upon the seed is germinated, the epicotyle, the baby plant, it is being cut, and then the scion of the selected variety is being grafted on the rootstock. So both the scion rootstock and scion are of obtained from the epicotyles only. So these two are attached to form a new plant. So this is very easy to obtain the union, because the plant is very small. So that is the reason why the union is much more important, and this is easy to some extent for the taking care for the resistant rootstock and the scion cultivar, which is of commercial importance. Both can be united and quickly the union can be obtained in case of epicotyle grafting. This is example is also mango. The fruit crop mango can also be propagated through epicotyle grafting. Then comes top working. Here we have grafting like cleft grafting as well as wedge grafting. In case of cleft grafting, the main tree or the rootstock is headed back, and then it is divided in the bit in center, and then scions, not only one, two or three or four can be inserted. The scion sticks can be inserted into the rootstock of the main branch, and then tied. Later on, the union can be obtained. Then the other type of grafting is wedge grafting. So we are making in the form of V shape, and here cleavage is seen on the rootstock, and then this is inserted, and the union is formed after few months. So this is the type. These are the different types of grafting. And if you look into the examples of horticulture importance, the veneer grafting is followed in case of mango, veneer or side grafting, as I told you. Then whip or tongue grafting. In the first picture I have shown you, like this. So this can be attached like this. This is followed in case of temperate fruit crops. Then cleft grafting, just I've been giving for examples for top working, like in case of mango, hazelnut, pecan, grape and all. Then wedge grafting, so V shape, this is also used for top working of old trees. Then bitch grafting is also repairing for damaged wood. So the damaged wood in case of temperate fruit crops like apple, pear, and cherry. So this the bark is removed, and then the scion varieties are inserted in case of bridge grafting. Then apical grafting, I have been telling you, mango and jackfruit are the commercial methods of propagation for these fruits. Then softwood grafting is also followed in case of cashew nut and mango. Then inarching, mango, jackfruit, sapota. So the scion variety, the rootstock is taken to the mother plant of the scion variety. Thereby, inarching is also most earliest method of propagation in case of most of the tropical fruit plants like mango, banana, sapota, custard apple, and all, loquat, lychee. Then we have double working. So as I told you, the wedge grafting and cleft grafting can be used to repair or damaged old trees. So rejuvenation of old orchards, double working is also done with the help of bridge grafting or cleft grafting or wedge grafting. Then comes budding. So the last type of vegetative propagation is budding. So budding is a deviation of grafting. It is wherein only one bud or a piece of bark with or without wood is used as scion material here. This is also called as bud grafting. Here you can see the major difference between budding and grafting. Budding is single bud is used as scion material, whereas in grafting is more than one bud is used. That is, a scion stick is used with more number of buds. Then the best time for budding and grafting in our conditions is for evergreens, the spring season is best for budding, and for grafting season, it is the rainy season where the humidity is high. Then in case of deciduous plants like temperate fruit crops, the spring season is best for budding, whereas winter season is best for grafting. See, if we look into the different possibilities of budding and grafting, here there is a possibility between the varieties within a same species or within a genus among the different species like plum, peach. They can be either used as a rootstock or scion variety. So Prunus domestica, on Prunus domestica, Prunus persica can be used on Prunus persica, Prunus americana, almond, like this and all. Then among the family, different genera can also be budded or grafted, like in case of pear, Pyrus communis, then Apis malus species. So among the different families also it is possible. Then in citrus also, trifoliate oranges, citrus can be can be made as rootstock, and upon the sweet oranges can be used as a scion. Then the different types of budding, I'm not going into the details of this budding. Just have a close look of the pictures here. Inverted T budding. This is a simple method. The first one is T budding, and the second one is slight variation is inverted T budding. So here you can see the T budding, preparation of the scion, removal of the leaves and all this. Only one auxiliary bud is present. That one is removed with the help of the bark. See here, it is retained. A small shoot is also retained, and the axillary bud is present here, and the rootstock is prepared in such a manner. The bark is removed in the shape of T, and then it is cut open, and then the bud is inserted, and it is allowed for union for few days. So thereby we can achieve T budding. We can achieve the union of the rootstock and scion. This is called T budding, which is very popular among all the budding. Then in some cases, inverted T budding is also used. The T here we used like this. The cut is made like this. In case of inverted T budding, the cut is made in the reverse T. So here the two flaps of the bark is open like this, and the bud is inserted on the upper direction only. Then you can see patch budding. Here you can see the picture of patch budding. Here a small rectangular piece or patch from the bud stick or the scion is removed. That is called as bud patch, and it is inserted into the rootstock. So for the rootstock also the same piece of bark is removed, showing the cambial tissues, and this is placed in the place of rootstock, and then it is tied, whereupon the union takes place. So this is ring or flute budding. So the bark on the rootstock, the rootstock as well as the scion, certain 15 centimeters to about 10 to 15 centimeters, the bark is removed on the whole of the stem, and in both the scion and the rootstock, and then inserted to the rootstock. This scion bud is inserted on the rootstock and then tied. So this is either flute or ring budding. Here you can see chip budding. So here, like in case of chip, and a small cut is made like L shape, inverted L shape on the rootstock, and then the scion also of the same size. The scion bud is inserted on the rootstock and then tied. So this is nothing but chip budding. So here you have seen all different types of budding. Then we'll go the examples, the commercial horticultural examples where budding is practiced is first shield budding is practiced in rose. Shield budding is tea budding. It is also called as tea budding. So it is rose and apple, pear, peaches, most of the temperate fruit crops, cherry, then sweet oranges are commercially cultivated. Then rose is also best example among the flowers. So because it is a perennial crop. Then among the patch budding, jackfruit, onla, mango, jamun, and chestnuts. Then chip budding, as I've told you, small chip is inserted. It is popular in case of grapes and also apple and pear. Then ring budding in bear, peach, and mulberry. Modified, a slight deviation of the ring budding is guava in case of walnut, pecan, etc. Then we have forked budding, like fork. It is followed in case of mango, cashew nut, and jackfruit. So we have learned about different methods of propagation until now. Seed propagation as deviations of the vegetative propagation. So all the rhizome suckers, bulbs and all we have seen on other side. And the other side we have seen cuttings, layers, budding and grafting. So which is the best one? So can we say which is the best one? Maybe we can tell for horticulture importance. If you look into the propagation by seeds, which is a major methods in which plants reproduce in nature normally. So we also know this is a widely used method for propagation of cultivated crops, either agriculture or horticulture. But seeds are readily available, inexpensive and easily to handle. But including vegetable crops, it is suitable for agriculture crops. But if you see the considering merits and demerits of both the methods, particularly in case of fruit crops or perennial plants, the vegetative propagation is more preferable than seed plants. And then through seeds, propagation through seeds, because of uniformity and precocity, because the clones which are obtained from the mother plants are uniform or true to type for the desirable characters, and they also bear early. So we should say that in fruit crops, vegetative propagation is much better, and to some extent for flower crops also, vegetative propagation is better for commercial propagation, whereas in seed propagation is best for vegetable crops and to some extent flower crops also. And vegetative propagation is good for plantation crops also, which are perennial in nature. They are trees. Here, for your exam point of view, I have made this table which shows the different types of propagation materials. You have to remember this and the different types under these propagation methods and the different examples of horticulture plants. This is important to remember. So cuttings, we have seen the different types of cuttings: hardwood cuttings, semi-hardwood cuttings, softwood cuttings, root cuttings. So the examples here are mentioned. You can go through and please remember this, the different examples. Then grafting, we have different types of grafting. Different fruit crops can be propagated by different methods of grafting. So the mango itself can be grafted by, can be commercially propagated by 3 different methods of grafting, and it depends upon it depends from region to region across the country. The farmers or the producers will follow different techniques. In South India, some techniques are followed. In North India, some techniques are followed like that. Then budding varieties, tea budding, patch budding, ring budding. So tea budding is most common in citrus, like that you have to remember. Then layering, tip layering, blackberry, grapes, lemon, simple layering, mold or stooling for rootstocks, air layering for ritchie, like that. Then runners, we have seen strawberry, suckers, date, pineapple, banana. Then separation or by division through combs, tubers, combs and bulbs. Ornamental plants as well as vegetable crops, then tubers and rhizomes and all this. Please take care. Please remember. So there is another method of vegetative propagation which has come into the commercial propagation of plants, like whether it is agriculture plants or horticulture plants, that is micropropagation. We have seen until now we have seen under vegetative propagation we have seen different variations which occurs naturally or artificially, but this is also artificially only, but which has gained importance in the recent past, that is micropropagation or tissue culture or in vitro culture. Until now we have seen in vivo culture. So some of them are within the plant itself, and in vitro culture is refers to the multiplication of the plants in under aseptic conditions. Aseptic means germ-free conditions on artificial growth media from plant parts like meristem tip, or the axillary tips, then callus, then group of tissues, then embryos, anthers, axillary buds. All of the plant parts can be used for micropropagation. So this is also similar to vegetative propagation or clonal propagation, like in case of layering, budding or grafting. This yields true to type on disease-free plants. Can be regenerated from a minute piece of plant in under aseptic conditions or artificial growth medium, as I have told you, rapidly throughout the year. So throughout the year, it can be possible to multiply plants in vitro or under artificial growth conditions. So this method is gaining popularity in the recent past because of the advantages over other conventional methods of propagation. So if you look into the layering, grafting and all, they need at least 3 months to form a unity, and then they can be separated into individual plants. At least 3 to 6 months we need time. But for this large scale, even though if we take time, we can produce large numbers in lakhs. For example, we say in lakhs. Here we can propagate them in hundreds or so. Here we can propagate them 1,000 for the same period of time, lakhs or thousands for the same period of time. So that is why it is gaining popularity in some fruit crops. It may not be possible in all the horticulture plants. It is commercially important in some fruit plants. So let us see the advantages one by one. It is for large scale of multiplication in lesser space and time. As I've told you, it produces virus-free plants. Then year-round production of the plant. So this is not the season is not going to hinder the production of plants, whether it is summer or winter. Either cases, we can go for production of the plant. Then tissue culture methods particularly effective in plants who do not breed from seeds. Seeds are not viable, male sterile, or not available, like parthenocarpic fruits like banana, pineapple, and in plants where propagation by conventional methods are expensive or tedious. like in case of orchids. So all this, under all these conditions, micropropagation is very advantageous. And in some fruit crops like papaya, where the production of female plants, dioecious fruit plants, is possible only through micropropagation. In that case, micropropagation is very much useful. Then comes the limitation. There are some limitations under this micropropagation also. If you see the cost involved in setting up and maintenance of a laboratory. So because this is in situ, it is done under laboratory conditions. The cost, initial investment is much more high when compared to other methods of plant propagation, and may not justify their use in all horticulture plants. So the plants which gain more returns in markets, then only we can go for plant propagation using tissue culture. Tissue culture techniques require skilled manpower, so normal labor cannot be done because this is the art and science of multiplication plants under in situ conditions, laboratory conditions. So all this has to be skilled labor. Then so a slight infection may damage the entire lot of the plants. So you need to be very careful while under all the steps, necessary steps should be taken care of. Then some genetic modifications or mutations of the plants may also occur, develop in some varieties and culture systems, which may alter the quality of the produce. Sometimes it may show on the genotypic and phenotypic characters of the plant which are produced under tissue culture systems. Then the seedlings grown under artificial conditions may not survive when placed under normal environmental conditions. So sometimes the hardening, this process is called hardening. Some plants, after removing from the tissue culture labs and growing them in the field, the success rate may not be, the success rate of the plants which are going under the fields may not be as 100% as perfect, but it is going to, some of the plants may die under normal conditions. So these also have to be taken care of. Then if you look into the different stages of micropropagation, it starts from 0 to 4 different stages. First one is identification of mother plant. Like in case of grafting and budding, the Sion variety need to be propagated, or the variety which have all the desirable characters need to be identified. Then the selection and processing of the explant. So which part of the plant is going to reproduce in the on the artificial media. So that should be whether we call it as explant. Whether it may be, as I've told you, it may be apical meristem or bud or anther or any other plant which we can cultivate on the lab. Then shoot proliferation. Then the other step is first when the explant is exposed to the growth medium, then first shoots will develop. Then artificial rooting has to be done for that shoots. Then the final stage is the hardening and acclimatization. Here you can see the plant tissue culture procedure. So the initiation stage, the selection of the mother plant, then removal of the explant, collection of the explants. So if you are taking just leaf or the apical meristem, then putting it on the growth media. Then multiplication stage would be the second stage. Then sometimes the shoot formation might be first compared to root formation. The third one, shoot formation, then root formation. Then after that, acclimatization in the polyhouses. Then thereby transferring them to pots or by directly transferring them to the fields. So the plants transferred to the nursery first, and then they're transplanted to the main field. And here this is a table which shows the different categories of the plants or the crops of horticulture importance, and the examples of the crops which are commercially cultivated through tissue culture. Among the fruits, Musa species, that is banana species, ranks first. Then Vitis vinifera, ananas, cosmosus, fragaria, strawberry, then sapota. Then not crush crops. Then spices, ginger, turmeric, vanilla, cardamom and all. Then medicinal plants, aloe vera, pelargonium, stevia, neem, pogostemon, all this. Then we also have a group of ornamentals like gerbera, dianthus. Most of the fleshy plants can easily propagated. Apart from monoecious, dioecious plants, monoecious plants perform well under tissue culture conditions like dianthus, anthurium. All the orchids are mostly cultivated through commercially cultivated through tissue culture only. Then we have cymbidium, all this syngonium, polyanthus, tuberose, and all this succulents can also be easily propagated through tissue culture. Then we also have some of the perennial trees, Tectona grandis, all the flowering trees can also be cultivated through tissue culture. So after going through the different types of propagation by seed, sexual or asexual propagation, we also have another term like apomixis, which is a deviation which occurs naturally in plants. Let us know what is meant by apomixis. Apomixis is derived from two Greek words, apo meaning away from and mixis meaning a mixture of, act of mixing. So this was first discovered by Leeuwenhoek in case of citrus seeds during 1719. So this scientist Leeuwenhoek have observed this apomixis, which is found naturally in some of the fruit plants. That is, especially he discovered in case of citrus seeds. What is this apomixis? This involves the formation of seeds without syngamy, that is, fertilization of the male and female gametes, and all the seeds are genetically similar. Therefore, it can be used as vegetative propagation. There, in sexual method of propagation, we have seen the formation of seed by the union of male and female gametes. But apomixis is a deviation of this. Here, the seeds are formed without the formation of zygote. We can simply tell that without the formation of zygote, the seeds are formed. And this term is called as apomixis. And this case, deviation is called as apomixis. Here, you should know one more important thing, that apomixis and parthenocarpy are asexual modes of reproduction of plants which occur naturally. But if you look into the parthenocarpy, which is a formation of the fruit without fertilization, whereas apomixis is a formation of seeds without fertilization. As I have told you, without the formation of zygote, the seed is formed and thereby the plant is produced. Thereafter, the plant growth is seen. Whereas in parthenocarpy, in case of banana, pineapple, and fig, where the formation of fruit is seen without fertilization, the seeds of or tinode may or may not be, and the apomixis is the deviation, the formation of seeds without fertilization. In the natural flow of biological processes, pollination is the first step. Let us understand in the first lecture, let us remember in the first slide I have told you how the normal growth cycle of the plant occurs. Like the pollination is important. This is the first step for the formation of fruit and seed. And the cells, upon pollination, then fertilization and cell divisions are important steps for the formation of the zygote. Then where meiosis occurs, the diploidy will come into haploidy. And the fertilization of male and female the gametes and the fusion will develop into zygote, thereby embryo, and the embryo thereby into the new plant. So parthenocarpy examples I am telling you, which is seen in pineapple, banana, cucumber, grape, watermelon, oranges, grapefruit, pear, and fig, etc. Then we also have some of the different types of apomixis. The entire process is cut short and seed formation occurs by the way of apomixis. So there is no formation of zygote here, but the seed is formed. That is the magic here. The seedlings produced through apomixis are known as apomictic seedlings. Then it can be of two types. So majorly the plants can be of sporophytic apomixis or gametophytic apomixis. The plants can be either occur, the seeds can be obtained from diploid sporophyte or haploid gametophyte. Let us know into the details now. Then based upon the reproductive behavior also, apomixis can be of two different types. The first one being obligate apomictic plants, where in those plants reproduction is only through apomixis. So and any other deviation will not be there. So those type of plants are called obligate apomictic plants. And the second one is facultative apomictic. So sexual reproduction also occurs in addition to apomixis. So apomixis itself is formation of seed without fertilization, and then those plants are obligate. They only reproduce through the apomixis. And whereas in case of facultative apomictic, the sexual reproduction also occurs along with apomixis. So majority of the horticulture crops comes under this category. Then if you see, I told you sporophytic and gametophytic. One of the deviation of apomixis, one of the types of apomixis is adventurous or nucellar or polyembryonic. So this is in majority of the fruit plants. This is a major type of apomixis which is found in horticulture plants. That is adventitious apomixis or embryonic, nucellar embryonic or polyembryonic. So this is of a sporophytic in nature. So here you see embryo develops directly from the vegetative cells of the ovules, such as nucellus, integuments, or chalaza. So you know the plant parts, the organs of the, this is the pistil, and here the ovary, and this ovule will be there. Before fertilization or the formation of gametes, the ovule contains parts such as nucellus, integuments, and chalaza. And directly from this parts, the seed is formed. There is no development of the embryo sac. However, the seed is formed. This type of apomixis is also called as sporophytic budding. So this is one case of sporophytic. They directly formed from diploid sporophytes, and they are of the plants obtained are of diploid nature only, 2N. So examples here are citrus and mango. Here you can see it is of a facultative apomictic. Both the seed is formed by zygote as well as the seeds are formed from the apomixis also, either nucellus, integuments, and all this. So the seeds of citrus have both the qualities. Even in the mango also, one single seed have all the different embryos also, apomictic embryos and normal seed-ring embryo. Then this is a flowchart which shows the different types of apomictics. If you look into the normal sexual cycle, so what happens in the normal sexual cycle? So say if you look the plant which produces diploid somatic cells. So both the male and female cells are of somatic in nature. If the seed is obtained directly from these somatic cells which are of diploid in nature, we call it as apospory. Then if the megaspore mother cell, so before meiosis occurs, like this is of 2N, after meiosis they becomes haploid. Both the male haploid gametophyte, male gametophyte as well as female gametophyte, the mother cell, for both the male and female cells, the megaspore mother cell, so we call it as diplospory. So this condition where the seed is formed from the megaspore mother cell of either anther or ovule, and the deviation is called diplospory, and the type of apomixis is called apospory. When after then meiosis occurs, so meiosis is the reduction division. So from N to 2N to N, that is haploid, we call it as apogamy. So the apomixis, we call it as apogamy. The first one is apospory, directly from the vegetative or the somatic cells. Then megaspore mother cells, which are of diploid nature, that is called diplospory. And the third deviation is, we call it as from the haploid cells, male or female, we call it as apogamy. So directly the seeds are formed from the haploid egg cells or the other cells of the embryo sac, like we have seen before in case of nucellar embryonic. And we also have other term like parthenogenesis, which now I'll give the details of this. So broadly, they can be divided into 4 different, as I've told you before. Apospory means some vegetative cells of the ovule in the embryo sac without meiosis develop into embryos directly from the diploid egg cells. In case of apple, Malus species. So of horticulture importance, and under some of the vegetable agriculture crops also it is seen. Then diplospory, here you can see the embryo sac is produced. Here there is no embryo sac without meiosis. So there is no development of embryo sac. Here embryo sac is produced from the megaspore mother cells, which may be of haploid nature or diploid nature. So before meiosis or sometimes after meiosis also it occurs. Apomixis occurs. Then the third third method of apomixis is apogamy. This is the origin of embryo from either synergids or antipodal cells, which inside the embryo sac. So after the formation of embryo sac, the deviation is either from the synergid cells or the antipodal cells. The seeds are directly formed. This can also be either haploid apogamy or diploid echogamy. And this diploid echogamy is seen in alien species and flower crops like iris species. Then the fourth type of apomixis is parthenogenesis, which is the development of embryo from the egg cell without fertilization. So this also mimics nucellular embryony or polyembryony, but here sexual formation is not formed. So there in nucellular embryony, as I told you in case of citrus and mango, both sexual as well as apomictic seedlings are formed or embryos are formed. But here in parthenogenesis, there is no sexual reproduction, there is no formation of embryo, but the embryos are formed without the formation of zygote, but the embryos are formed directly from the egg cell without fertilization. This can be of haploid parthenogenesis or diploid parthenogenesis. So haploid is Solanum nigrum and diploid example is Taxus. So but horticulture importance is with the help of this adventitious polyembryony only. This is of major importance. This comes under this is commercial importance, and you have to remember for exam point of view. Then there are also several advantages and limitations for this apomixis. See here, the first one is fixation of heterosis in the hybrids. So we can fix the heterosis like the plants, the hybrids which are of premium quality when compared to the mother plant. So the characters can be fixed with the help of this apomixis. Then easy maintenance of the pure lines, purely in case of developmental stages. So plant breeding, it is very much important, this apomixis. Then synchronous flowering and maturity of the crop. The nucellus seed hybrid varieties is conveniently maintained as a hybrid varieties. Then minimum isolation distance in hybrid seed production. This is achieved through apomixis. Then pollen dispersal is not relevant during hybrid seed production. So the pollination, the tedious method of pollination, these are all not required in case of apomictic seedlings. Then, however, limitations are also there. Apomixis is a very complicated process. If you look in case of breeding methods, then estimation of the level of facultative apomixis is tedious and time consuming. Then the proportion of sexual progeny is affected by environmental factors like day length and temperature. So abiotic and biotic factors are also important if you are going for breeding through apomixis. Then in the absence of morphological markers linked to the apomixis development, maintenance of apomixis stocks becomes difficult. So we have to link these morphological markers with DNA markers also, so that it becomes easy. But however, it is difficult. So it might become a limitation for this breeding purpose also. Then the genetic basis of apomixis is not clear in most cases. So all these are the different types of means of sexual propagation as well as asexual propagation of horticulture plants. Thank you. That's all for today's class. We'll meet again for the next class with a different topic. Thank you.